In this talk, we will learn about the different mechanisms that give rise to species. The formation of species requires the population to evolve. We can define evolution as changes that occur in populations over time, and these are due to changes in the genetic composition or genotype of individuals in the population. The genetic changes in organisms of a population can occur randomly. There are different factors that can promote genetic variation in a population, which include mutations, gene flow, mating patterns, etc. Having genetic variation in a population is important because it aids certain organisms in the population to survive and reproduce better than others under appropriate conditions. This allows the population to evolve and ultimately for species to form. There are two main ways by which populations can evolve. The first one we'll look at is anagenesis. In anagenesis, the population is undergoing changes. However, members of the population are still able to breed with one another, and hence the evolution is taking place in a single lineage of organisms. On the other hand, cladogenesis is when one lineage is able to split into two or more populations. These two or more populations no longer breed with one another, and hence cladogenesis results in the emergence of new species. There are two main ideas that have been proposed to explain the pace at which populations evolve. In the case of gradualism, the species that are formed diverge slowly from the ancestral organism over long periods of time. And thus, in every generation, there are subtle changes, and thus, with many generations, we're able to see more and more divergence of two species that arose from a common ancestor over time. Unlike gradualism, where we are seeing a slow divergence of the species over long periods of time, in the case of punctuated equilibrium, the species are rapidly formed, and we can see distinct differences amongst the individuals between different species very early on in the process of species formation. Once these species have been formed, there are extended periods where there's not much change happening in the individuals belonging to those species, and these periods are called as stasis. Now, it's perfectly reasonable to assume that different organisms have either gone through the process of gradualism or punctuated equilibrium as the species were being formed. The process of forming species is called as speciation, and species can be formed by different mechanisms. These include allopatric speciation, peripatric speciation, parapatric speciation, sympatric speciation, and the more recent artificial speciation, where human activity is involved in the speciation process. In this talk, we will focus more on allopatric and sympatric speciation. The concept of allopatric speciation was proposed by Ernst Mayr, who is the same biologist who proposed the biological species concept. In the case of allopatric speciation, a geographical barrier splits a population into two or more groups. This barrier can be in the form of mountains, oceans, rivers, etc. The barrier prevents gene flow between the groups because they're unable to mate with one another due to the presence of that barrier. Hence, the individuals present in these groups evolve independent of one another. there's more and more genetic differences that are between the individuals of the different groups, and these genetic differences arise due to mutations, natural selection, genetic drift, and other factors. Now, what happens if these groups meet later? Well, that depends on how much time has passed. If the groups are able to mate before any reproductive isolating mechanisms have evolved, then the groups are able to interbreed with one another and gene flow resumes between the populations. This causes no new species to form and it results in more genetic variation among the population. On the other hand, if the groups meet after reproductive isolating mechanisms have evolved, then depending on the kind of reproductive isolating mechanisms present, there can be different outcomes. If prezygotic reproductive isolating mechanisms have evolved, then there will be no gene flow between the groups because no zygotes can be formed when we have individuals belonging to the two groups mating with one another. In this situation, the two groups now have become two different species. On the other hand, if not enough time has been passed for the evolution of prezygotic reproductive isolating mechanisms, however, post-zygotic reproductive isolating mechanisms have developed, then we do have a situation where individuals from the different groups can mate. And when they do mate, there is a possibility for them to generate hybrid progeny that are not viable. They could also produce hybrid progeny that are sterile, or they can produce hybrid progeny that are fertile. However, the successive generations of these hybrid progeny lose their fertility. Thus, in these situations, there is a reinforcement of the reproductive isolating mechanisms by natural selection, and eventually prezygotic reproductive isolating mechanisms will evolve that will allow now the different groups to become their own species. Another way by which species can be formed is through the process of sympatric speciation. In this case, new species arise from a population in the absence of a geographical barrier. Sympatric speciation has been controversial among different scientists. In the case of sympatric speciation, reproductive isolating mechanisms evolve within a single interbreeding population. Since this is a special case by which species can occur, it is thought that there have to be special circumstances that allow sympatric speciation to occur. Disruptive selection, which is a type of natural selection, can favor sympatric speciation. In the case of disruptive selection, there is a certain event that occurs. For example, the introduction of a new predator that can cause a change in the population such that the two extreme phenotypes that are shown, which in this case is small body size versus large body size, are selected. The individuals that show these extreme phenotypes tend to breed only with one another and eventually can evolve to form their own species as time passes. Another way sympatric speciation can occur is through polyploidy. Polyploidy is normally seen more commonly in plants than in animals. Let us take the example of two species of plants wherein one has 60 chromosomes while the other one has 62 chromosomes. Both of these plants are diploid and hence they both have homologous sets of chromosomes. Both of these plants are able to form gametes, and let's imagine one forms the male gamete that now has 30 chromosomes, and the other forms the female gamete that has 31 chromosomes. It so happens that these gametes are able to fuse to form a zygote. The zygote thus formed has 61 chromosomes, and let us assume that it is viable. But we should note that even though it has 61 chromosomes, we are not able to see the corresponding homologous chromosomes. Now, this hybrid can still survive, however, it cannot undergo sexual reproduction because there's no homologous sets of chromosomes. We can, however, envision a situation where non-disjunction occurs. And in this case, there is a duplication or replication of each chromosomes during the S phase. However, mitosis doesn't occur, and thus we now are able to form cells that actually have 122 chromosomes, and in this scenario, every chromosome has a homologous pair. Now, in this situation, the plant has the ability to undergo sexual reproduction and is now able to generate progeny with genetic variation. Now that the gene flow can occur among the progeny of the plants, a new species can arise. With this, we come to the end of the talk where we learned about the types of evolution that occurs in population, namely anagenesis and cladogenesis. We also learned about the concepts that explain the pace at which species form, which is gradualism and punctuated equilibrium. We also learned about the different types of speciation and focused more on allopatric speciation and sympatric speciation.